This is part 3 of the Chapter 5 lecture notes. The previous slide we were discussing channel proteins. So let me explain to you the difference between channel and carrier proteins. A channel protein just has an open pore, an opening that allows an ion or a polar molecule to move through it. But if it's a carrier protein, it's going to bind to that substance, change shape, and then when it changes shape, it basically opens up to the other side. And so in quotes, it says it will carry it to the other side. It basically just opens up by changing shape and allows it to move to the other side. Many carrier proteins allow movement in either direction depending on the concentration gradient. One important carrier protein is our glucose transport proteins. So glucose, I guess, in this example is represented, even though glucose is normally a hexagon, in this example, the glucose is represented by the little oval-shaped, little green oval-shaped molecules. So glucose is polar, so it cannot diffuse through the phospholipid bilayer. It does have to diffuse through a channel or a carrier protein. And in this case, you can see that the concentration of glucose on the outside in the extracellular fluid is high, and the concentration on the inside is low. So glucose is going to move down its concentration gradient from high to low, and so it's going to move into the cytoplasm by binding with the carrier protein, as you can see here, and then the carrier protein changes shape and opens up on the bottom end, allowing the glucose to come into the cell. Now, osmosis is a special type of diffusion. It is the diffusion of water across a membrane. There are a couple of ways to remember the direction that water moves. First of all, you have to understand that water, in these situations, water is the solvent, so there are solutes dissolved in water. Now, the solutes can be sugar or salt or a waste like urea, or they can even be oxygen or carbon dioxide gas. But some solute is dissolved in water, and that solute is too large to be transported across the membrane. And there are no channel or carrier proteins for that solute. So instead of the solute moving from its high to low concentration gradient, water is going to move instead, because the movement of water can change the concentration of the solution. So I always remember that water moves in the direction of the higher concentration. Water moves toward the highest solute concentration. And so water follows solute. So if you just look at the concentration of the solute in the different solutions, water is going to move in the direction where there's a higher amount of solute. So in this case, the water on the left is going to move to the right side of the beaker across that membrane, that semi-permeable membrane that is only permeable to water, that will not allow the solutes to move. And when it does, the level of water rises on the right side of the beaker and it lowers on the left side until you can see the space between those purple molecules is about even. So the concentration of solute molecules in the water is fairly even on both sides. So the water is going to follow the solute until the concentration gradient, or the concentration gradient doesn't exist, basically, until there is no difference in concentration on either side of the membrane. So here it says water moves from an area of higher water concentration to lower water concentration. This is another way of looking at it. Where do you have the highest water? You have the highest water on the left side. So water is also going to move from where water is high to where water is low. And that's another way of looking at it. But the reason I teach students that water follows solute is a lot of times what you have is an example where you have concentrations listed. Like you might have a beaker. So here's your little beaker with a spout. Here is your semi-selectively permeable membrane. And then let's say on the left side, you had a 1% concentration of solute. Let's just say it's salt dissolved in water. And on the right side, you had a 10% concentration of salt. Okay, so if you remember water follows solute, water's going to move from the left to the right because it's going to move to where there is the highest concentration of solute. But you can also just kind of reason to yourself that if the concentration of solute is 1% on the left, then it's 99% water. And on the right, it is 90% water. So you can also look at it as water is going to move from where water is high to where water is low. In any case, either way that you look at it, water follows solute or water moves from higher water concentration to lower water concentration, either way it works. I just find that students remember water follows solute a little bit better. Now, tonicity refers to the concentration of the solution that the cell is surrounded by. So if the solution is hypertonic, that means that the concentration of solute outside the cell is higher than inside the cell. If the solution is isotonic, then the concentration of solute outside and inside the cell is equal. And if the concentration is hypotonic, the concentration of solute outside the cell is lower. So when we look at tonicity, oftentimes we use the example of red blood cells to show what animal cells do when they're placed in different solutions. So if an animal cell is placed in a hypertonic solution and water follows solute, hypertonic means that the solute concentration is higher outside the cell. So if water follows solute, water is going to leave these red blood cells. The red blood cells are going to shrink and shrivel, which is not healthy for the cells. In an isotonic solution, the red blood cells will maintain their shape because there might be water coming into the cells, but the amount that comes in is equal to the amount that leaves. So there's no net gain or loss of water in the red blood cells. If you place red blood cells in a solution where the solute concentration is low, that would be a hypotonic solution. An example of a hypotonic solution would be just plain distilled water with nothing dissolved in it. It would have a 0% solute concentration. So if you place these red blood cells, which have a higher solute concentration, I can't write it because it's still red, but the red blood cells have a higher solute concentration. So water is going to move into the cells and they're going to swell, and more than likely they are going to burst. So of course that's not healthy for the cell. Plant cells are a little bit different. They behave a little bit differently because plant cells have a cell wall, and that cell wall does not change it's rigid and it does not change size. But inside the cell wall is a plasma membrane, and the plasma membrane is represented by this green line. The cell wall is represented by the black line. So the thick black line is the cell wall, and then the thin green line is the plasma membrane. In a hypertonic solution, which has a higher concentration of solute, so you can think of it as just like really salty water, like ocean water, for example. So you place a plant in salt water or you water it with salt water, then the plasma membrane is going to shrink away from the cell wall because the plant cell is going to lose water. Water is going to follow the higher solute concentration. Water follows solute. So the plant cell the plasma membrane is going to shrink away from the cell wall. And what you would see in the plant is the plant would wilt, and it would probably kill the plant. In an isotonic solution, if you water a plant with a solution that contains some solutes, but not just, like tap water is good. It's going to contain some solutes, but the same amount of solutes that's inside the plant cells. You're not going to have a change in the shape or the size of the plant cell. In a hypotonic solution, so if you water the plant with distilled water, which has a lower solute concentration, then water from the outside is going to move into the central vacuole of the plant. It's going to cause the plant cell to swell, but it will not burst. So what we say is that in a hypotonic solution, the plant cell develops turgor pressure. And that's what we see when you water a plant and then the plant kind of stands up straight and tall and pretty and healthy looking. But if you water a plant with salty water, it's going to cause plasmolysis. And what we see the plant wilting. And this is an example. On the left, without adequate water, the plant will lose turgor pressure and it will wilt. And you can restore that turgor pressure by watering it with a hypotonic solution, because water is going to enter, and it enters from the roots, I know, but it's going to enter the plant, and the plant cells will individually swell, but they won't burst because of the plant cell walls. Now, another thing that can cause wilting is to water the plant with salt water, and that will cause the same thing. But this plant is wilted simply because it's dehydrated. This one is just a plant that needs to be watered. Here is an organism that you can find easily if you take a drop of pond water and look at it under the microscope. This is an organism called a paramecium, and it's from the domain Eukarya, because it does have a nucleus and organelles, and it's from the kingdom Protista. So it's called a protist. It lives in fresh water normally, and so what happens is inside of the cell has a higher solute concentration, and the fresh water that it lives in has a lower solute concentration. So the fresh water is hypotonic to the inside of the paramecium. So what's going to happen is water is going to continuously move into the paramecium. So what it does, so that it doesn't swell up too much, is it has a contractile vacuole that pumps the water out of the cells so that they do not burst.